welcome everybody to what is now our 26th Inspiration Exchange session. For those of you who don't know me, my name is Fergus Imrie and I'm a postdoc in the Schaar Lab, and I'm hosting today's session alongside Mihaela and Tennyson Liu. Today's session is about revolutionizing clinical trials with machine learning. And I'm delighted to announce that today's session will form mainly two parts. In the first part, one of the PhD students from our lab, Alihan, will give a presentation on when to make and break commitments. And then in the second half of the session, we'll be moving to a roundtable discussion followed by Q&A with several experts from industry who are involved in the area of clinical trials. And I'll introduce our four guests more formally during that part of today's session. Throughout today's session, if you have any questions for any of our speakers or panelists, please ask them in the Slack channel, and we will try and address as many questions as we can during the Q&A portion of the session. Without any further ado, I want to hand over to Alihan for his presentation on when to make and break commitments.
As Fergus mentioned, I'm going to present you a new paper that will appear in ICLR, and it's called When to Make and Break Commitments. And the paper is very much, has applications in other domains, but it's very much about portfolio level management of clinical trials when clinical trials are seen a kind of commitment. And I want to start my presentation with first highlighting that the area of clinical trials is full of interesting machine learning problems that can potentially be addressed by many different fields of machine learning. These problems include ones that are encountered during the planning of trials, as well as their conduct, the analysis of their results, and how their results can be used in actual clinical practice. And today, the paper I present will just be an example of how reinforcement learning can be helpful in conducting clinical trials. More specifically, I will talk about how we can ensure that clinical trials can target the right patient populations. But why study clinical trials, especially from a machine learning perspective? Well, the first reason I think is that there's a huge potential to do really meaningful impact on how medicine is practiced. Trials are very costly and they take a lot of time. A typical trial costs upwards of $30 million to run, and they are usually finished within 1 or 2 years. And despite this huge cost and commitment required, more than 1,800 trials are run per year, which kind of highlights their importance to the evidence-based medicine. And you can imagine that any kind of improvement that is made to this process would have a huge impact on the efficiency of how these trials are run. And second important reason, I think, this is coming more from a machine learning perspective, is that I think the constraint setting of clinical trials poses really unique reinforcement learning challenges. The problem that I'm going to present today is called optimal commitment problem, which is a new type of optimal switching problem that we define, is an example of this. And although the motivation very much comes from the clinical trials, we will also see that this problem has applications in other areas like portfolio management and energy networks. So considering these clinical trials with a machine learning lens can lead to solutions that are more generally applicable, because it presents this very unique kind of trade-offs that need to be addressed. And I should also mention what is the current state of how clinical trials are run. So the gold standard for showing the efficacy of a new drug is a randomized controlled trial where patients from a population are recruited and they're assigned randomly to a control and a treatment group. And then their responses are compared against each other to see whether the treatment that is being tested has any positive effect. And there are well-recognized problem with this paradigm. First of all, these trials are very hard to adapt as they collect more information. Usually all aspects of the trial design, such as how many samples that are going to be collected, need to be specified before the trial before the trial starts. And secondly, usually these trials target only one type of population, meaning they have a set inclusion criteria, and this inclusion criteria is not adapted as the trial continues. And this can be problematic in certain scenarios. Here's a motivating one for the problem that I'm going to describe. Suppose there's a treatment that is effective for a particular subpopulation within the whole patient population, but when you average effects over the entire population, the treatment turns out to be ineffective. But of course, without running a trial, we have no knowledge of this. So if we follow the standard approach and run a randomized controlled trial that targets the overall population, since the overall effect is negative, it will be really likely that this trial will fail. And as a result, the treatment would be denied for the subpopulation that it actually would have benefited. So this is the kind of the scenario that we want to avoid. Of course, to kind of address this, many adaptive experiment designs are proposed. So to start with, I mentioned already that RCTs are kind of never adapted and they only target an initial patient population. So the common approach to addressing this is adaptive enrichment designs. In these designs, there is a designated fixed decision point and data is collected up to that point. And then the results are evaluated to decide whether the rest of the trial will continue targeting the overall population or whether the focus will shift into a fixed subpopulation. And there are extensions of this called adaptive signature design. Again, there is a fixed decision point that is planned beforehand. But now, instead of just deciding between a subpopulation and an overall population, the goal is to kind of determine a continuous criteria that can describe any kind of subpopulation. And again, related to this, there are randomized clinical trial approaches that feature futility stopping. Notably here, the decisions are made in a sample by sample basis. But usually it is only limited to a go versus no go decision, meaning do I continue my trial as it was planned, or whether I stop because it seems highly unlikely that the trial is going to succeed. And what I'm going to propose, which is called optimal commitment, kind of tries to combine strengths of both of these approaches. First of all, optimal commitment can make decisions in a sample by sample basis and not just during fixed decision points, but also it's not limited to go versus no go decisions. It can find which populations should be targeted among multiple populations that are provided. So then what is optimal commitment problem? It's a new type of optimal stopping and switching problem that aims to model this situation and address that. So kind of continuing with the clinical trial example, suppose we have an experiment that has a certain desired size and it is launched. And what will happen during this trial is that up to a specified sample horizon, we will see patients arriving one by one. We will see which population these patients belong to. And after assigning a control treatment or a new treatment, we will see what the outcome associated with them are. And at the end of our sample horizon, we run a hypothesis test and the trial is either successful at showing efficacy or not. And until the last sample is observed, since the samples are random, kind of it's uncertain whether the trial will be successful or not. And this trials like this have certain characteristics. First noteworthy thing is that there are usually continual costs associated with each sample. This could be, for instance, like time spent recruiting a single patient or time spent on treating one particular patient. And after each sample, we are kind of have to pay this cost, which I denote with minus here. And another characteristic is that there is an uncertain reward, and it's only received if the trial is followed until its end. That's why I call this a commitment. To receive a positive reward at any point, you need to commit to finishing the trial. And a final aspect is that it's not usually possible to change this design on the go. Then the question optimal commitment problem asks is, given a time step, meaning I'm in the middle of the trial, maybe I collected 50, 100 samples, whatever. How can I decide between continuing the trial as usual, stopping the trial completely, or potentially switching to an alternative trial? And here the trade-off is really between, if I suspect my trial to kind of fail, stopping would obviously be advantageous because I would avoid all future costs that I need to pay. But of course, if I were to continue my trial, I can collect more information that will make me more certain about whether the trial is going to be successful or not. And there's a balance that needs to be achieved between these two when making this decision. So this might have all sounded a little bit abstract. That's why I want to highlight that you can think of optimal commitment problem as a specific instance of a reinforcement learning problem. First, the states here consist of the active design that is currently being followed. This design might dictate what is the targeted sample count, what is the targeted population, and so on. And there are mean outcomes associated with each population that is considered. And the transition function here is very simple. When I take an action, i.e. when I choose what trial design that I want to follow, the active trial design becomes that design. And then, as I run these trials, my observations consist of pairs, where I see a patient, where I see which population a patient belongs to, and the outcome that is associated with that patient. And my observation function is dictated by the active trial design that I'm following, meaning the population from which the patient arrives, for instance, depend on what is the design target population is. And my outcomes depend on the mean outcomes that is specified for each subpopulation individually. And then the kind of the decision-making problem that I described earlier can be summarized as a reward function. In this reward function, I always pay a cost for each sample I'm seeing, and I only receive a reward if two conditions are both satisfied. First, I committed to a trial until its designed number of samples are all collected, meaning my actions didn't change for a well-defined period of time. And second is that at the end of this time period, the data I collected is indicative of a success. So this is an alternative way of describing the optimal commitment problem. Of course, the language I used was heavily influenced by this clinical trial example, but you might consider other commitments in other domains, and this formulation could potentially be beneficial for those as well. One example I want to give is portfolio management, where similar to a trial, maybe an asset you invest in takes time to build an additional value, and you need to commit to that asset until then. But at the end of that period, an asset might end up losing value, similar to how a trial might end up failing. And of course, staying invested in, as said, as an opportunity cost, which you are paying continually. And another good example is energy networks. Suppose I'm trying to decide which power plants to turn on and off. And of course, switching a power plant is not an instantaneous thing. I need to commit to decisions. And when doing this, I'm trying to match moving energy demands. If I don't predict this accurately, I might make a wrong commitment. And of course, a plant, while it's on, consumes resources, which is equivalent to a continual cost you are paying. So optimal commitment could be useful in all sorts of domains. So then the next question is, how do we solve it? Before I delve into that, to be able to provide better insight, I want to highlight an even more simplified instance of the optimal commitment problem, where there is only a single commitment. In the case of clinical trials, this would mean that I only have one design that targets one specific population, and the choice I need to make is between stopping that trial or just continuing until its end. And my observations might be sampled from a Gaussian distribution with an unknown mean. This corresponds to the effect the treatment has for that subpopulation. And supposed to be able to make decisions, I'm keeping track of a moving empirical mean, which I denote with mu. And this is the same reward function that I shared earlier. For each time step I'm continuing with the trial, I'm paying a cost minus C. And at the end, potentially I can gain a positive reward R. I gained that reward only if I didn't stop until the trial was finished, and only if the trial was successful. Here, I assume success means the empirical mean reward that I see is larger than 0, which corresponds to a positive effect. And as I said, the first instinct to kind of attack this as a reinforcement learning problem, because it is an instance of a reinforcement learning problem. And the way you would do that is kind of try to find the optimal value functions. That is, given a time step, given that I see a specific test statistic, a specific mean empirical outcome, what is the value or the total reward when all the costs are subtracted that I can achieve? And here is a very simple case of the problem where the sample horizon is just 4, the reward to be gained is 10, but each time step costs 1. And here are the optimal value curves for all the time steps leading up to the end of the trial. And if you, to understand them better, you can focus on the first graph. So here, as the mean outcome I have tends to more negative and negative values, it means that the trial I'm running is less and less likely to succeed. Hence, in the limit, my optimal value to gain becomes 0, because I now know this trial is certain to fail, and I just need to stop. I do not gain any rewards anymore. And on the other hand, if you look at values of mu that get larger and larger, my trial becomes more and more likely to succeed. At some point, it's almost certain that it's going to succeed. And at that point, the value to be gained is the reward that I'm going to receive at the end, in this case 10, minus the cost of samples that I would need to pay. There is 3 more time steps to go, so I will pay a cost of minus 3. So the limiting value here would be 7. There is 2 things to highlight here. First is that these value functions are very clearly non-convex functions. This is important because the existing methods for solving reinforcement learning problems like this usually rely on convex function approximators. So those solutions would not be feasible in the case of optimal commitment problem. And a second observation is this. As you can see, when mu gets larger and larger, there is a threshold where our value function becomes positive. So that threshold is very significant because if our test statistic is ever below that, we would want to stop the experiment now. But if it's above that, meaning the value is positive, there is reason to continue, and we would continue our experiment. The difficulty is basically calculating what this specific threshold is. And we can look kind of into more insights that can help us specify some characteristics of this threshold. And interesting baseline to consider while doing so is what I call the greedy approach. Kind of a natural policy to follow might be to look at the expected outcome of a trial and decide based on that. With a certain probability, the trial is expected to succeed. I multiply that with its reward and subtract all my future costs. That's my expected kind of outcome that I'm going to receive from my trial. And if it's negative, I stop. This I call the greedy approach. And very similarly, this is again kind of like a thresholding type of situation. Under a certain threshold, when your test statistic is under a certain threshold, this would be the case and you would stop. And when we compare the optimal threshold and this threshold needed for making greedy decisions, we see that the optimal threshold is more optimistic in the sense that it kind of is biased towards continuing the experiment. As time steps move on, I can look at my test statistic, and if it's ever above this red line, both the optimal policy and this greedy approach would stop. And if it's below this blue line, both the optimal policy and the greedy approach agree that the trial should continue. But in between, there's this yellow region where while a greedy approach would tell you to stop, the optimal decision is actually to continue. And the reason this gap exists is that continuing a trial is less of a commitment than stopping it entirely. If I ever stop my trial, that's the last bit of information I will ever see, and I will have no chance to reevaluate my decision. Whereas if I decide to continue now, I don't need to commit until the end of the trial, and I can just see one more time step and kind of reevaluate my decision. And the fact that I can reevaluate my decision has value. And due to that value, this kind of yellow gap between the two approaches exists. And if I want to behave in an optimal way, I should be optimistic. And kind of an intuitive way to continue this argument is that I need to be increasingly less optimistic, because as the trial goes on and I get closer to the final sample, there will be less opportunities to reevaluate decisions. Hence, the value of the fact that you can reevaluate will also decrease. You can consider a trivial instance, for instance. Right before the last sample is collected, in these graphs, in this example, it's time step 17, because the sample horizon is 18. That's the final decision you are making. There won't be another time step to reevaluate anything. So in that particular case, this gap becomes 0, and you are making the same decisions as the greedy approach. And so these are some of the characteristics these thresholds have. And based on that, what we do is kind of propose a heuristic algorithm that is optimistic and in fact increasingly less optimistic. And we call that algorithm bias OCP. The idea is kind of simple. You are maintaining a Bayesian posterior over mean outcomes of every population. And at each time step, by doing a combinatorial search, you are identifying a candidate experiment that you can run instead of an ongoing experiment. This is basically like determining what subpopulation, what narrower population I could be targeting instead of the ongoing trial and the target population of that ongoing trial. And once a candidate experiment like this is determined, what Bayes OCP does is that it compares the expected utility of the candidate experiment to the ongoing experiment. The greedy approach would be that whenever candidate experiment has a higher expected reward, we would go with that. But instead of doing that, Bayes OCP behaves optimistically, and it looks at the probability of the posterior probability of the candidate experiment having a higher value. And if it's larger than, say, 80%, only then it decides to switch to the candidate experiment. So that might have been kind of a lot to follow, but in a more intuitive sense, what Bayes OCP does is that it requires stronger evidence in favor of candidate experiment to ever prefer it with respect to an ongoing one. I want to quickly show some basic results to show that this works. So I consider simulated trial environments. And if you remember the motivating scenario, I kind of consider when that might happen. Here, the green instances, here, there are two subpopulations. And in green instances, the treatment effect is effective for all of them. So in these instances, you will expect the randomized clinical trial to run well. In amber instances, the treatment is not effective for the combination of two subpopulations, but it is effective for at least one subpopulation. So this would be the motivating scenario at the start of the presentation. And this is where we expect Bayes OCP to do better. And a red instance would be where the treatment is not effective at all. It negatively affects both subpopulations. And indeed, what we see that if you look at the amber instances, or also the average results across all instances, Bayes OCP tends to perform the best because it achieves the best balance when deciding when to stop and when deciding when to target a different subpopulation. Kind of the conclusion of this method is that first, one observation that we made was that general purpose reinforcement learning is not always sufficient in finding solutions to particular healthcare problems. Optimal commitment problem was just one case of this. While most existing solutions were infeasible, by carefully modeling the problem and analyzing what is the structure of an optimal solution might be, we were able to find a much stronger heuristic algorithm. Kind of a second takeaway message is that whenever making decisions, possible information gain must be accounted for. So machine learning often very frequently focuses on making good predictions, but this problem kind of demonstrates that even if you have good posteriors of mean outcomes, you kind of need to still think carefully about how you are going to incorporate them into your decisions. And the fact that reevaluations might happen could provide value, and those need to be accounted for. And the kind of the synthesis of these 2 points is that we need more ML solutions, like optimal commitment problem, that model unique aspects of healthcare problems or problems in other domains more directly and more specifically, instead of trying to give general solutions to all decision-making problems. And second, these models need to consider uncertainty in a principled way because they have an important effect on how we make decisions. And if you want to read more about the paper or our other work on clinical trial design, here are some QR codes.
Without any further ado, we're going to move to our panel discussion, and I'd like to introduce our 4 guests today. Our first guest is Sean Hayes, who is an associate principal scientist at Merck Co in New Jersey. Secondly, we're joined by Carl-Fredrik Burman, who is a senior principal scientist at AstraZeneca. He's also an associated professor at Chalmers University of Technology and a guest researcher at the Karolinska Institute. We're also joined by Suetonia Palmer, who is a professor of medicine at the University of Otago in Christchurch, New Zealand. And we're also joined by Miro Dudík, who is a senior principal researcher at Microsoft Research. Today's panel is going to be chaired by Professor Mihaela van der Schaar. So Mihaela, please take us away with the panel discussion.
Yeah, thank you very much, Suetonia, Cafe, Sean, and Miro for joining us today. Very big pleasure to have all of you, and you have so diverse backgrounds, and yet so many related works of yours are related to what Alihan was talking about. Suetonia, as the clinician in the Zoom call, I'm going to start with you and ask you, what do you see as the future of clinical trials in general? And do you think that machine learning can help us empower this future of clinical trials, and in what way?
Perfect question, because I think clinical trials desperately need to be transformed by machine learning. There is absolutely no question. So COVID enabled us to disrupt trial technology into ways that have made it much easier and much quicker to bring trials to completion. So Alihan showed us the 33, the huge costs and the amount of time. But I think the improvements with COVID have been only incremental. They've enabled us to shorten them or focus on them to some extent, but they haven't been transformative. And I think we desperately need almost instantaneous trials that patients don't even really need to be part of. So I think that's the aspiration. And Alihan and your lab's work get us so much closer by being able to truly know, once we've started the trial, when exactly we can stop and not make the wrong decision. So I think the future of trials is actually machine learning to a great extent.
Well, I'm quite happy to hear your positive look on that as a machine learner. Sean, as a machine learner working in the industry, in pharmaceutical industry, how do you think, which elements of clinical trials you think will be most impacted by machine learning and why? Where do you see the biggest opportunities in this area?
Yeah, it's a tough question. Just, I mean, there was the initial slide, right, of all of the different areas. I think all of them are very impactful. Even, I mean, just setting the machine learning part aside, I mean, the impact of quantitative methods for decision making, which broader than just machine learning, but machine learning has a lot of new methods to offer. I think there's been a lot of movement in that direction. Just Merck particularly. A lot of our commercial success, Keytruda's approvals were very much down to selecting the right subpopulations, the right clinical trial design. So I think with our company, it's been fairly well recognized. But seeing some of the presentations like this, there's a really far way to go. I think some of the the adaptive type designs, things where we're getting more flexible about the way that things are designed at the decision making, just because currently organizationally, like there's sort of some separations between the people actually running the clinical trials, some of the ways they're designed, and then the people with the quantitative skill sets doing that. So it's I wouldn't have necessarily even thought of clinical trials in some of the ways that you're kind of proposing these optimization problems. So I think it's using the decision making to be more embedded in the design of the trials. There's a tremendous amount of improvement that can be offered there, I think. Yeah.
Cafe, as somebody that has worked on the clinical trial designs for many years, both in academic environments and industrial environments, more from a biostatistics point of view, do you see opportunities for machine learning, and where do you think machine learning can join forces with biostatistics to really provide new answers and new ways of thinking?
So I think what's hot right now is digital twins. And Mihaela, may I pose a question to Alihan? So a great presentation. I think that having the investment perspective and also seeing the patients and that we may need personalized treatments. But then you have a lot of different subgroups. And I wonder, could you instead, as an extension, perhaps have covariates in describing these patients? And could you use machine learning to learn how these factors will impact the response, and in that way gradually use ML to find the right population? So it looks very similar to what you did, I think.
Yeah, absolutely. So like in this case, the kind of focus was very much on the decision of switching between a candidate experiment and an ongoing experiment. And how that candidate experiment was determined kind of assumes that you are provided a set of discrete subpopulations beforehand, and you can only combine them. But this was only because that wasn't the focus. So that there could be kind of any process that you want there that kind of analyzes covariates more kind of like in a machine learning way to come up with that candidate trial, right? And then you can incorporate it into what has been presented here, for instance.
So I've been working previously on the very simple problem that we have two subpopulations, biomarker positive and biomarker negative. But then I'm thinking that if we're using machine learning and have a set of covariates, you could gradually within the trial see that a subpopulation, not really a subpopulation that's a set of X's, but more that in a high dimensional space, you will see that a subspace of the patients are likely to have a positive effect. So I think that you could probably do kind of the same thing, but use more of machine learning and borrow information between different parts of the total population. So I think there's
that sounds like homework for Alihan Cafe. Thank you for that.
Yeah.
Maybe I can, do you want to respond just very briefly, or can I move on to the next question that I have actually for Miro?
No, I absolutely agree, yes.
Miro is a machine learning researcher in reinforcement learning, and of course, Microsoft is looking at this area of clinical trials as well. Which parts of reinforcement learning, of the reinforcement learning toolkit, do you expect that are going to be most applicable in the context of clinical trials?
So, do I think about this? There are kind of two different ways how reinforcement learning can be injected or kind of like combined with the idea of clinical trials. We can either think about some kind of adaptive treatments. So when the disease itself is perhaps evading, or there is some kind of notion of habituation, and so when the treatment itself is adaptive, and we call that policy in machine learning, that's such an adaptive treatment. So that's one area. The other area, which I think like today's talk was an example of, was really using some kind of an algorithm, or some kind of like a more algorithmic experimental design, adaptive experimental design to drive the trial itself. And I really liked actually Cafe's point because basically like a lot of my work was in the area of contextual bandits, which is exactly kind of the approach that's used for personalizing user experience. You can have from recommender systems, etc., where we're in a situation where we have a range of covariates describing the user, and we're trying to figure out like what content to present to the user, for example. So here in this instance, it will be the choice of treatment available. And there are some limitations, because my sense is that often in clinical settings, the signal about quality of our action, in our words, of the quality of the treatment that we took at that point in time is much delayed. And so this is a huge challenge for some of these kind of existing machine learning approaches, just kind of that it might take a while before we know. but there might be still some interesting application areas, like thinking about like maybe compliance, where you might get like a more immediate immediate signal about like whether the participant in the experiment is following the protocol, and something that could be still used to reduce the cost in a slightly different way. So yeah, I do think that there's many kind of opportunities ranging from personalization, which to something more like adaptive treatments.
Before I move on, I want to ask you one more question, Miro. Are there any lessons from applications of reinforcement learning in the tech industry, in particular in Microsoft, but more generally, that do you think would be applied to clinical trials in particular, which are really a quite different area, especially clinical trials for drug design or for medical interventions?
Yeah, no, I think my sense is that that it's important to think about the settings that where personalization might be helpful versus the settings where more like kind of A/B, basically like the design is more useful, you know, and so I feel, and this also that's kind of like where we in industry we saw that we cannot just kind of throw these contextual bandits and everything. That sometimes it's really just kind of like the careful, like thinking through like what's A, B, and C is really important, you know, rather than just kind of trying to personalize everything. So that's one lesson. And the other one, which I think is kind of interesting, which is kind of this kind of design of perhaps something like overlapping, kind of overlapping studies, you know, where you perhaps are, you could be when appropriate, you know, so we need to think through all of the kind of Belmont, you know, accord continuous, like of the beneficence and justice. But when we are thinking through things like these overlapping designs might allow us to basically use the data better and ultimately possibly benefit our population better. So I think it's slightly speaking to what Suetonia was mentioning. We're just kind of enrolling people, especially in situations where the standard of care is somehow lacking, you know, like the threshold for enrolling in one or multiple studies could be, you know, might make a difference.
Related to that, Suetonia, Shan, and Cafe, how important you think in a clinical trial is the target population, this inclusion criteria to be well defined for the trial success? And more generally, what are the considerations when deciding which population to target for a particular clinical intervention? Can I, Shan, Suetonia, Cafe, feel free to jump in.
Yeah, I've got an easy answer. Just because, I mean, so Merck right now, you know, as a company is majorly, you know, defined by Keytruda as a cancer immunotherapy. Early on in that, we were in a race with Bristol Myers Squibb, and the approval of Keytruda over BMS's drug was down to a subpopulation selection. I think it was actually one of the early cases of actually an approval based on a specific cancer biomarker. So I mean, it's, you know, the entirety of Merck as a machine right now is down to that kind of thing. I think that excellence in clinical trial design is a huge focus of the company, and it's you start talking about even just things like getting the number of subjects down by tens, and people get really excited. So I mean, it's maybe not as exciting of a problem as some other, when you start talking about discovering new therapies and some of these other things. But I mean, in terms of actual, real world effect, and the amount of money that it saves, how much additional development that allows you to do, just what it does. I mean, it's, the entire engine of drug development right now is held up by the cost of clinical trials. So it's absolutely massive.
Perhaps I can add a bit to your example. So immuno-oncology has been tremendously important. It was the Nobel Prize in Medicine a couple of years ago. And especially these PD-L1 drugs has had enormous success. I think that saved the life of Jimmy Carter, if I remember correctly. An enormous impact for many, many people. And what Merck was doing then, I think regulators are typically telling us to go with a wide population because their question, and I have been pressing them on this, and to my surprise a bit, they have been more concerned about having a too narrow population and saying, how do you know that the drug isn't effective in the biomarker negative population? So often we're going with the wide population, but the statistical testing is often targeted into the right population. And in this, for immuno-oncology drugs, different companies have gone with very, very different levels of PD-L1 expression. So if Merck is getting this right, then you get the drug, you get an approval, whereas someone else is not doing this right and they fail in the trial. So this is hugely important. And then in exactly that setting for PD-L1 drugs, there have been research also, because in early trials, we tested PD-L1 drugs versus chemotherapy. And then chemo was better in the beginning. And then PD-L1 drugs was better after a longer time period. So it was a lot of ML work on trying to figure out which patients would die early or have a progression early. So it's better to go with chemo. So it's not only the PD-L1 expression, but it was also work into a lot of different covariates and much more into the ML space.
Yeah.
Can I add? Yeah, absolutely. So in my field, we're not rich in biomarkers. We don't plan populations. My field is diabetes and kidney disease. And we've got a number of examples where extremely large trials have demonstrated an average effect and have led to significant clinical changes in guidelines. For example, statins and also anemia drugs, where when we look in retrospect, we identify that there were very significant parts of the population who either received harm on average or certainly no effect. And so the guidelines, the clinical guidelines recommended treatments that were actually ineffective. So that's one point to make. The second point to make, I think, is that we often exclude people from our clinical trials because we don't think that we can include them because they're not going to be adherent to the treatment or they're very hard to find. I'm thinking particularly older people, people with lower socioeconomic equity, and also women are frequently very lowly represented in clinical trials. And so I think knowing our populations and ensuring that all people get access to trial interventions and that we understand the effects in a broad range of people is incredibly important for expanding access to medications and also providing good outcomes for a broad range of people.
Thank you so much for that. Maybe a final, yes?
Yeah, may I pose a question to Suetonia as well? So in diabetes, we have half a dozen classes of treatments. So do you really think that that we should be doing what we're doing now? Don't you think that we could find subpopulations that will go different paths about which drug to start using? So for example, sulfonylurea, it's have efficacy going down over time. Is this really the right choice for all the people that get it now? Or do you think that we can have different types of diabetic patients with different optimal treatments?
I think we can. I think we can personalize diabetes treatment much, much more effectively than we are. A really good example is whether the newer treatments, GLP-1 receptor agonists and SGLT2 inhibitors, have different effects in people with different levels of kidney function, and whether those benefits are preserved even with people with very low kidney function who are currently excluded from those trials. I also think we need to be able to use observational data and electronic health records in a much more impactful and effective way so that we don't have to wait for clinical trials, but we can understand individualized treatment effects in data that we already have. And so that enables us to be much more instantaneous in our understanding of treatment effects, and also enables us to test those effects in a broad range of people instantaneously who are in the electronic health record but are not in the clinical trials.
Well, that's music to my ears because we are very much working hard to try to develop technology that hopefully will be useful to do this analysis from electronic health records and hopefully create control arms that could be useful or at least identify subpopulations. And I think overall, I was delighted to hear all of you thinking about the fact that machine learning hopefully can complement, not substitute, but complement the world of biostatistics that has made powerful impact in this area. And hopefully by joining forces, we can make a difference. In view of time, I have many questions to all of you, but in view of time, I'm going to leave it at that and give the microphone back to Fergus. Maybe there are questions from the audience.
Thanks, Mihaela, and thanks to all of you, to everyone on the panel. I think that was super interesting. We do have a few questions that I'd like to take in the time remaining. The first one I think is for potentially any of the panelists to answer from Ashish. Do you want to unmute yourself and ask your question? Ashish, are you? Oh,
yeah. So I was, first of all, loved the talk by Amin and also the panel. I had a very generic question in terms of actual new innovations reaching real world clinical trials. Where do we think are they most likely to these innovations to come from? From academic medical researchers, academic ML researchers, statisticians? I was just curious. Any of the panelists?
I'm not sure who'd like to take that.
Maybe I should jump to say
go for it, Mihaela.
It's my belief that this is not a topic that just one group or one type of discipline can really contribute. Actually, it's my biggest belief that by joining forces in between biostatistics, statistics, machine learning, hopefully we can come up with innovative approaches. And while the academia, I think, can play a role, I don't think that academia alone can make the disruptive impact that needs to be happening, because this is a very complex, highly regulated area where industry has been active for many, many years. So while we can propose approaches, I think that it is only by joining forces with industry that we can really make the impact needed, given the complexity of the area. What I am very enthusiastic about, though, is we have a lot of interesting work that has been happening in reinforcement learning. Miro here mentioned contextual bandits and many other areas that our group also was active on, and there is a lot of interesting machine learning work in this area for many years. And I'm happy that hopefully now we are at a level of engaging with clinical trials and hopefully making an impact there. So I think that it's about a collaborative approach. At least this is what I think. I don't know what the others think, but.
Mihaela, I'm just going to take us. Yes, I totally agree, Mihaela. As a pure academic with no industry involvement, I'm profoundly impressed and interested in the work that industry is able to provide. Ashish, I also think that we need to examine a number of existing medications, not just new medications. There are so many treatments out there that actually have no evidence of benefit or potential harm that we need to go back and use our new technologies to be able to examine all over again.
Especially in oncology. I've heard that a lot of oncology drugs, as long as they don't do harm, but they don't provide significant benefit, the FDA is quite conservative at approving them, right? Or like, it approves drugs which may not have a significant benefit, but as long as they do no harm, they get approved. So, yeah.
Ashish, I think sometimes it's down to what outcomes are being measured and how effectiveness and efficacy are actually understood. And with new technologies, we can go back to some of those outcomes and reflect and refresh the information that we have about existing drugs. And also, as the team have already said, understand the effects in subgroups of populations to understand whether there are people who do get benefit and so that those drugs are really still very, very beneficial for a group of people, but may not need to be used in other people who may have been in the original trials.
Thanks, Suetonia, and thanks, Ashish, for your question. I think we've got time for one more very quick question. And I see Richard Dearden has a couple of questions for Alihan, but maybe if you could just ask one of your questions to Alihan, Richard.
Sure. I wasn't sure if you're going to take questions to the or just to the panel, but I guess I was interested in the impact of what you're doing. Sorry, I have the camera in the wrong place. The impact of what you're doing on the actual sort of clinical trial power itself, right? Because if you're normally clinical trials, we don't do too many intermediate analyses because we worry that because it depowers the trial, then you need to recruit more patients in order to be sure that your multiple hypothesis testing isn't screwing things up. And it seems like what you're doing is basically doing an interim analysis after every patient, which seems kind of problematic. I'm wondering if there's a way to avoid that, that depowering of the trial.
Yes, there's certainly a depowering here, but I want to mention that here the multiple hypothesis testing issue is not at each sample, because what this method does is that it views each trial as a black box, and we use those analyses to decide on which trial to follow, but these analyses are not part of the hypothesis testing at all. Where the depowering happens is that, for instance, during my design, my algorithm switches 2 times. That's effectively equivalent to running 3 trials, right? So that becomes equivalent to 2 analysis points or like 3 hypothesis tests. That's where the depowering happens. So it's
basically where you change your decision are the only times you're doing the equivalent of a, right? That makes sense. Thank you.
Great, thank you, Richard. And I want to thank all of our panelists again for the panel discussion. I'd like to turn over to Zhaozhi to give an update on SynCity, which is our group's recent library for synthetic data that we released a couple of months ago.
I'm very excited to make some updates on SynCity development. In March, despite the turbulence of world economy, we continue the development of SynCity, and we managed to ship out several quite important and highly requested features, including the addition of a new data augmentation pipeline, which allows users to perform a one-click benchmark on different synthetic data augmentation technologies. And this can help users to find the correct model class to address issues in data scarcity and the representation or transfer learning setting. In addition, we have implemented the diffusion model for generation. We have brought in the TabDDPM model, which is a new SOTA benchmark in tabular diffusion model. And we've also made various other improvements in tutorials, documentations, bug fixes. And we thank again for all the community contributors for their wonderful contribution. So coming next month, there will be, we will continue to add more plugins and metrics, including some of the recent cutting edge study that were accepted in AI Stats and iClear this year. We will be also incorporating more fairness metrics. In addition, we'll continue our developer program. So please, if you contribute significantly to a SynCity package, we are very happy to provide reference letters and other acknowledgements for your contribution. And we've also created the Slack channel in this Slack workspace that is dedicated for SynCity discussion. And last but not least, we are hiring. There is an open position in Cambridge Center for AI in Medicine, and we are looking for motivated software engineer. Please reach out if you want to become an expert in cutting edge machine learning and AI algorithms, contribute to open source software, and most importantly, make impact by addressing pressing issues in healthcare and medicine. You can find more information about the job description and application link in the link below. Thank you.
So just to very briefly wrap up, for anyone that wasn't able to make today's session that you know might be interested, today's session will be put on YouTube soon. So please do let your friends and colleagues know about today's session. The next Inspiration Exchange session will take place on the 18th of April, and more details will follow shortly. To stay up to date on Inspiration Exchange, do visit our dedicated website and sign up on that website to ensure you don't miss any notifications about Inspiration Exchange. Finally, a massive thank you again to our presenter, Alihan, and our 4 panelists, Sean, Miro, Cafe, and Suetonia. Thank you very much for joining us today, and we hope to see you again soon. Bye-bye.